her passion for science trickled over into passions for other things, and she poured herself equally deep into those.
She was a super famous molecular geneticist, but she also was this super passionate conservationist.
She was quite a gal. I have never known anybody else like her.
Hello, everyone, and welcome to The Sound of Science, the podcast highlighting the voices behind the breakthroughs at Oak Ridge National Laboratory.
We're your hosts, Morgan McCorkle
and Jenny Woodbury. We're doing things a little differently this episode. We're looking at the career and legacy of the late Liane B. Russell, one of ORNL's most renowned scientists.
A genetic pioneer, a conservationist, a mother. Liane Russell was a force.
And if you're not familiar with her name, chances are you'll be familiar with the scientific discoveries her work produced.
To tell her story, we've talked to a few of those who knew her best, and a few that she inspired.
And thanks to an interview from the Atomic Heritage Foundation archives, you'll also be able to hear some of it in her own words.
Liane, also known as Lee, was born in Vienna, Austria in 1923. She was one of 3 children. Her father was a chemical engineer, and her mother was a homemaker.
In 1938, she and her family fled from Austria shortly after Nazi Germany annexed the country.
And we're very lucky that we were able to leave very early. We left just 5 weeks afterwards and went via Prague and Brussels to London.
Her family stayed in London for 3 years before making their way to the United States in 1941.
We left before America got in the war because when we came here, there was no war here. You know, it was very strange coming from all the bombings and everything. But in December of the same year that we arrived, of course, Pearl Harbor happened. So it was a short time between wars, sort of.
Once in the U.S., Liane attended Hunter College in New York. Although her dad was a chemical engineer, Liane wasn't planning on becoming a scientist herself.
She actually wanted to be a writer during her first year of school, but eventually decided to pursue a career as a medical doctor.
Her path changed once again while attending a summer research program at Jackson Laboratory in Bar Harbor, Maine in 1943.
I really got bitten by a research bug. I was looking down a microscope and I saw a mouse fertilized egg, and I thought, my God, 3 weeks from now, less than 3 weeks from now, this is going to be a mouse, a complete mouse. And that really was an amazing thought. And that sort of got me turned on.
The summer program not only introduced her to her career-long passion, but also to her future husband, geneticist Bill Russell.
Liane and Bill became an inseparable team that would be world-renowned for their numerous discoveries in genetics.
After the Manhattan Project, ORNL, then known as Clinton Laboratories, ramped up its work to understand the health effects of radiation.
Research on radiation had been done with Drosophila, also known as fruit flies, but fruit flies aren't a good genetic match for humans.
And the research suggested radiation had little effect on the flies.
I guess an early idea was, we know enough. We know what it does to Drosophila. That's all we need to know. And other people like Bill and Lee said, no, that's not all we need to know. Drosophila eggs are not like human eggs and sperm. The whole developmental process is different, and we actually know nothing about what happens to human germ cells.
That's Dabney Johnson. She was a colleague and close friend of Liane's.
Germ cells are the sex cells, or eggs and sperm, that allow genes to be passed on from generation to generation.
Mice, unlike fruit flies, are genetically similar to humans, so studying their germ cell development would give more of an insight into the effects of radiation.
So the Russells came to Oak Ridge in 1947 and established a large-scale mammalian genetics program known as the Mouse House.
It was a health experiment, and the whole system was set up to be able to do that in huge numbers so that you would have statistically significant sample sizes for all of that. So for every million mice that you grew from parents who had been exposed to radiation or chemicals, you had to have 1 million mice whose parents had not been exposed so that you had the control group. So this was an enormous undertaking to get all the kinds of radiation in all the kinds of doses that it could be done in, and then plus check an awful lot of chemicals. So it was a huge experiment.
Dabney was a student, then colleague of Liane's in the 1980s.
At its capacity, the mouse house could hold 200,000 mice. We asked Dabney to paint a picture of what it was like to work there.
At one time they'd had several 100,000 mice in that building all at the same time, so you can imagine what it smelled like. And, those of us who went in there, you get so your nose doesn't recognize that smell. But people would walk in the door and go like this. They would cover their mouths or, get out a handkerchief and hold it over their nose. It was noisy. There were cage washing machines going on and people, and the acoustics were terrible. Things were echoing all over the building.
Over the next 6 decades, Liane and Bill made a number of groundbreaking discoveries with their research produced from the mouse house. A few in particular are almost common knowledge nowadays.
Did you know her research on mouse embryos produced the Universal Prenatal Guidelines for X-rays during pregnancy?
By studying the mouse embryos, she determined the critical periods in prenatal development.
That's why we all wear lead aprons when we get x-rayed for anything now, because it's very dangerous to very early embryos. So she would do that in mice, in pregnant mice, at different times during the development. And then you can, the gestation's 3 weeks for mice, 9 months for people. And you can translate those two timelines to say that if it's dangerous to mice at day 3, then it's probably dangerous to human embryos at week 6, something like that.
Remember when you learned about the X and Y chromosomes in biology class? Well, Liane was the one who discovered that the Y chromosome notes maleness in mammals.
The Y chromosome had been discovered, but its role was unclear in mammals.
So some of the work had been done in way lower organisms, and then some work was done in Drosophila, fruit flies. That was the major model animal at the time. And Drosophila don't have Y chromosomes. Their male determining things are scattered around on their other chromosomes. And so what do mammals have? And so she did some very complicated experiments that eventually, and soon after that, we learned how to stain chromosomes and actually count them. And sure enough, anything that had a Y chromosome was a male. A female who has one X chromosome is a female. A male that has 2 X chromosomes and a Y is a male. So if you have a Y, you're a male.
The Russells' work also helped set occupational limits for radiation exposure.
Liane retired from ORNL in the early 90s, but that wasn't the end of her work at the lab. She remained very active and continued to do research.
Dabney became the lead for the mouse house research, and Liane's boss.
Have you ever been in that situation where somebody who towers over you with their knowledge and their intellect is actually supposedly, under your authority? It was very crazy. I can remember trying to do her performance appraisal, and I just thought, this is completely ridiculous. But she stayed very much involved, very interested, still doing work, still publishing papers, still, gathering data, going to meetings and talking.
Liane's enthusiasm wasn't limited to genetics research. She and her husband were also passionate conservationists.
It started in the mid-'60s when some lands and waterways were being threatened here in East Tennessee, places that they loved, and I loved, going with them around places here in Tennessee.
That's Liane and Bill's son, David Ace Russell.
The couple was instrumental in founding the Tennessee Citizens for Wilderness Planning nonprofit organization, which helped establish the Big South Fork National River and Recreational Area, and obtained a National Wild and Scenic River designation for the Obed River.
It kind of naturally grew out of, I think, their devotion to science in general. My mother started as more of a humanitarian type. She was, I think, pretty well cultured in a lot of ways. And she originally intended to be a doctor and to help humanity that way. Then she met my father, and he kind of convinced her, I think, that a pure research role would also be very helpful. But she always took that basic idea of helping people, I think, into her science.
Liane passed away July 20th, 2019, but her legacy lives on at ORNL.
Research at the Mouse House ended in 2009, but the lab still does work in genetics, just not in mice.
Jerry Tuscan is the director of the Center for Bioenergy Innovation at Oak Ridge. He's also a geneticist who primarily studies plants.
Initially, it would seem there would be no connection between the work that Liane did in mouse genetics and what I've been doing in plant genetics. But at the DNA level, things are very similar and very common. Shared mechanisms for inheritance and how phenotypes or traits are passed on from parents to progeny.
While there are parallels in their work, the tools they use couldn't be more different.
In the late 90s into the early 2000s, Jerry was part of a team that sequenced the poplar genome using a computer program.
And if you're wondering what a genome is, it's all the hereditary information that's coded into an organism's DNA.
As the Russell's career was ending, the whole evolution of DNA sequencing was evolving, was just coming on the scene. And so we do things today in a matter of 24 hours that would have taken weeks of time. They just didn't have the tools, the mass spectrometers, the DNA sequencers that we have now. Literally, a postdoc in my lab can order a kit, isolate DNA in a couple of hours, drop the DNA into the kit, mail it out, and by the time he comes back or she comes back the next morning, has the results. And it's just phenomenal, the speed at which we can do things.
In addition to all those tools, Jerry has access to Summit, the world's fastest supercomputer, to help him sequence genomes.
So if you think about poplar having 40,000 genes and just the pairwise interactions, but genes operate in these protein complexes where there are 4 or 5 members of the protein complex. They all have to show up at the right time in the right tissue at the right moment to have some function to cause some reaction. It's been impossible to map that or to understand that. You can study one, you can study a half a dozen, but on a genome-wide scale, it up until about 2 years ago was impossible. So we were able to use Summit to predict gene-to-gene interactions called pleiotropy, to predict all pleiotropic interactions in the poplar tree, all of them, and in humans. And so for the first time, instead of trying to chase a gene to a particular phenotype, we're able to stand back and say, okay, it takes these 100 genes all operating in concert to lead to the point where you have the opportunity to express the phenotype. And so the evolution of our computational capabilities is really, I think, going to just launch us faster and further than we ever have gone in the past.
Research results from Summit are enabling scientists to tackle a range of problems, like developing better biofuels and bioproducts, to fighting cancer and the opioid addiction crisis.
Liane was a pioneer for women in science, and that legacy continues with the fellowship named in her honor.
The Liane B. Russell Distinguished Early Career Fellowship was established in 2013 with the goal of expanding opportunities for early career researchers.
And it is essentially for young women getting, their own independent research facilities, because so many women who were just as educated as some of the men ended up working as a partner or technician, or not a high-level technician almost, to somebody instead of doing their own research. And so this is one way of getting young women their independent facilities and opportunities. And they're not all biologists, they're everything. They're physicists, mathematicians.
And even ecologists. Melissa Kreger, whose work focuses on plant microbe interactions, became a Russell Fellow in 2015.
So I got to meet her right after my fellowship started with a couple of the other fellows. And it was amazing because she was an older woman, but she was still super sharp. So my fellowship research was focused on understanding how the loss of hemlock trees in kind of mountain forested systems, how that affected carbon and nitrogen cycling. And so she was really passionate about hemlock trees. So she proceeded to tell me all about the hemlocks in her yard and how she was spraying them with this insecticide to keep the adelgid at bay. And it was really important to her. She was very interested in the science I was doing because she had already known kind of everything that was going on with the hemlock trees.
To date, there have been 10 Russell Fellows.
It's hard for me to even fathom kind of what she went through on a daily basis. Even as a woman in science in 2020, I still often walk into rooms where I am the only female scientist in a meeting. But I have a lot of advocates and supporters, and, there are other women scientists that I can turn to now. So I can't even imagine what it was like for her to know that she was the only one. Like, she was one of the only ones there. She probably didn't have the kind of support. So the fact that she did everything that she did at Oak Ridge kind of paved the way for all of us to kind of continue on in her work and get to be scientists at a national laboratory.
As we've heard, Liane was a woman of many talents, but we did learn that she had one weakness: chocolate.
She was scrupulous about labeling foods on the date of purchase long before that was mandated by any authorities, and continued to do it afterwards, by the way. But she was scrupulous about that, except in the case of chocolate, which was, in her opinion, something that lasted forever. It didn't matter how old the bar of chocolate was that you found in the cupboard. It was good. So I guess she thought chocolate never expired.
This was a fact that was also well known among her colleagues.
So she always wanted fine chocolate. She wanted, Swiss chocolate and all these imported chocolates. And I can remember one time in the old mouse house, there was a vending machine. And for some reason, she was out of chocolate, and it was 4:00 in the afternoon, so there had to be chocolate. And a couple of us caught her buying ho-hos. I mean, they couldn't rot, they're full of such horrible things. And so she put her money in, and she turned around and she looked, and she knew she was caught. And we died laughing. And she just said, "I don't care if you know I'm eating ho-hos." And she had to have her chocolate.
From her love of chocolate and the natural world to her groundbreaking research in genetics, we hope you enjoyed learning about the life and legacy of an amazing woman.
Thank you for listening to this episode of The Sound of Science. Make sure you subscribe wherever you get your podcast from so you don't miss an episode.
And we'd love to hear your feedback, so leave us a comment or a review.
Until next time!